We have a special welcome here for the participant from Arkansas. I hope, Amanda, you're on. You're the only one from outside of Illinois. Thank you for being with us. One announcement. There is a forthcoming free program in a week's time, next week, and I did put that announcement on the left-hand side of your screen. If you haven't done it, open the bubble at the bottom left-hand corner. This is the same place you're going to put your questions, comments, discussions, and anything you'd like to suggest. In fact, at the end of the meeting, if you can tell us a few things, it would help. But we shall also send you evaluation form because we need your feedback. We need to know where to improve on this presentation. So next week, there is that program. You can register. It's also free. It's going to be presented by Zach Grant about extending the season. The other thing I would like for you to note or to hear about is that this topic that we are going to talk about today is extremely extensive and complex. It's really huge. So we are going to be touching on a whole lot of topics in there, but we're not going to go into depth. We'd like to hear from you if any of those topics are ones that you have an interest in, and we can pick on particular topics later to get into depth. But rest assured that we shall answer all your questions. I'm going to put our emails on the chat box, and you can write to us anytime hereafter, and you can be assured we'll get back with you. But as of right now, we get going because we need all the time we can get. If I can get this thing going. Give me a second here. I have lost the screen. Okay, just hold on. But anyhow, everybody's seeing the first slide, and our emails are there. And Lori, if you just go to the second slide.
It's there.
Where it says introduction.
It is up.
All right, thank you. So soil-borne pathogens are important. They are the unseen enemy. We never get to see them. The only reason we know they are there is when we see the disease on the above ground portions of the plants that are growing. And as you can see from that slide, you can have severe reductions or extreme reductions in productivity of the plants that you're trying to grow. They could be seedlings, they could be bigger plants, but you can have a limitation on the growth and productivity. And indeed, as you can tell from the National Agricultural Statistical Service, in 2007 they found that 1.6 million tons of crops on 250,000 acres worth 700 million suffered a 10 to 15% loss. And that's a big loss. And these are diseases caused by various pathogens. And we will know that when we say pathogen, we are talking about a disease-causing organism. That's what we are talking about. The fungi, the bacteria, the viruses, and the nematodes. And pathogens have evolved to live in the soil. And some will live for a short while, maybe a season or 2 seasons, whereas others will just stay in the ground forever. They are always available. A stressed plant will always be welcome for them. And we will talk about various management options that there are. There's a whole lot of them, some of them still undergoing research, but we'll mention them. Others have been shown to work, and I'll let Laurie take over from here.
Okay. Thank you, James. So we're going to be talking about several of these different types of pathogens that can be found in the soil. And the first one I want to start with are water molds and the downy mildews. The specific ones for these that are going to be you're going to be dealing with on a regular basis in your garden will be Pythium, which causes seed rot, seeds damping off, root and soft rots, especially with the fruits that are in contact with the soil. Phytophthora, such as late blighted potato. Don't see a lot of that down here in southern Illinois, but we do see that in other places. Fruit rots and blights. Also, we're going to talk about the downy mildew diseases of dicots, such as tomatoes and peppers, as well as the downy mildew diseases of monocots. So when we talk about the water molds, we're talking about the diseases that can occur because there's an excessive amount of water or the water that's being used has been contaminated. So the diseases, the two types, are going to be the ones that affect the plant parts that are present in the soil or in contact with the soil. So you got to stop and think about the seeds, any type of fruit that's sitting on the soil itself that may have that contamination occurring, the root systems of the plant. And the second type of plant disease we're going to talk about are going to be the diseases that affect only primarily the above ground portions of the plant. And all of the downy mildew, there are some species of Phytophthora as well. So we'll discuss that a little bit more in detail. So let's talk a little bit about the Pythium Phytophthora. The Pythium Phytophthora, and there are two others, if you're familiar with them, they're Rhizoctonia and Fusarium, can cause damping off. Damping off affects seeds or seedlings at an arbitrary age of 6-week-old seedlings. And that's generally going to be the upper limit of those seedlings for the plant age to get this damping off disease. Plants over the age of 6 weeks are generally infected in a similar manner, but they're said to have root rot or stem rot. Pythium is carried on the soil and plant fragments and in the water. Wet soil conditions can favor the growth of Pythium, and these are also called water molds. Pythium produces a fertilized reproductive structure, which then produces a thick wall surrounding it, and this is part of the resting stage or the overwintering stage. These are resistant to adverse temperatures and moisture, and it's the survival mechanism for the disease. Pythium species occur in surface waters and soils. They live on dead plant and animal materials and on fibrous roots of the plants. They always require water for spread of the disease. Phytophthora occurs when the soil is too wet and the soil temperature remains fairly warm, above 65 degrees Fahrenheit. The air temperature is in the 75 to 85 degree range, and humidity is high. Various Phytophthora species can survive cold winters and hot, dry summers, as either through the resting stage that I talked about earlier, or through mycelium or hyphae in infected roots or stems. Poor germination of seeds and poor emergence of seedlings is a direct result of damping off infections in the pre-emergent stage. Some of the examples I wanted to show, if you haven't seen it before, but these are some of the things you may find in the garden. As far as in the upper right-hand corner, that would be the root system. So what you're looking for in a nice, healthy transplant is going to be nice, healthy, white root system. Anytime you have this browning or dead roots, you've got a water problem. Down at the bottom, you have the Pythium, where it's going to be pre-emergent, where either the soil is too wet or too cool, and the seed is just going to rot in there. Some of the other things you may find in the garden as far as Phytophthora is some sort of wilting or dying of one or two plants, or some of the fruit that may be on your plant itself. The second one I want to talk about are the downy mildews. So the water molds do include the downy mildews. In Illinois, it is thought to overwinter in greenhouses or travels in or on cuttings that you may be receiving. The spores themselves can travel long distances by wind. Now this is becoming a problem in Illinois, especially with basil. They're seeing a lot of this in the central Illinois area. So keep an eye out in your garden if you notice you may have some problems. The pathogen for most downy mildews are carried in the seed or the bulb, or the infection takes place at the seedling or the young plant stage. It causes systemic shoot infection in the host plant itself. Most of the time, rainy weather When it's wet, the spores will germinate and they'll spread either by wind or water. They're also favored by cool temperatures, such as 58 to 72 degrees Fahrenheit and a relative humidity above 85% at the leaf surface. Older plants, when they're attacked, may develop small or large, localized infected areas, and it can spread into young tissues. It causes rapid and severe losses of young crop plants, often destroying them from 40 to 90% of those plants. The severity of the disease depends on the prolonged presence of wet, cool weather, during which the mildews sporulate profusely. They produce those spores quite a bit. Infection can decline when the weather becomes hot and dry. And I just wanted to show some examples of the water molds that you're going to see as far as the downy mildews. The one on the left is on the basil, so you'll see a little bit of yellowing that occurs on the leaf itself. You flip it over, you see those sporulations that are occurring on that basil. In 2015, Dr. Bavadous to the University of Illinois Extension found downy mildew on Illinois pumpkins, and we are the #1 producers of pumpkins in Illinois. So this is going to be can be a very catastrophic disease that will hit our fruits and vegetables. And then again, the ones that you see as far as the cucurbits. Let's talk about the soil-borne bacterial pathogens. Bacteria are single-celled microorganisms. They're very hard to see. About 1,600 species are known. Most of them help decompose organic matter every year, so a lot of them are very beneficial. About 100 of these species cause diseases in plants. Bacteria multiply very quickly and can produce tremendous numbers of cells in a short period of time. Bacterial diseases of plants occur in every place that is reasonably moist or warm and affects all kinds of plants. The agrobacterium or crown gall, Ralstonia, bacterial wilt, the xanthomonas, such as the blights or bacterial spots, the streptomyces, the scab of potato, and Pseudomonas, soft rots of the potatoes and onions, as well as some species or races of Erwinia, are all typical soil inhabitants. The streptomyces can survive indefinitely in moist soils, except in most acidic ones. So if you're growing some potatoes or something and it's fairly acidic, to remove some of that scab, that would be good. Xanthomonas and Pseudomonas usually persist in the soil for only a short period of time. Here are some examples of what you're going to see as far as Erwinia. If you haven't seen it in your garden, of course, in the upper right-hand corner is the streptomyces of the potato, which we can see on a regular basis for the common scab. Down at the bottom of the Ralstonia on the pepper. If it's going to be a bacterial infection, one of the good ways of trying to test for it to see if it is Ralstonia is to take a cutting of a plant, put it in a cup or a glass of water, such as they have here on the right-hand side. After a few minutes, you'll see the streaming that can occur from the plant cutting itself. If you see that bacterial streaming, there's a good indication that you may have a bacterial disease. So that's a good indicator if you have questions on that. Almost all plant pathogenic bacteria develop mostly in host plants as parasites on the surface of the plants, such as in buds, as well as in plant debris or in the soil. They can build up their populations within the host plants, but only gradually decline when they are released into the soil. So that means if you have susceptible hosts planted in successive years in your garden, you can build up the disease population in the soil because it's not removed after a season. It may take 2 or 3 years. So you need to keep that in mind. On some of the others, plants often survive, like I said, in buds, seeds, wounds, in the exudate or anything that's coming outside of the plant from inside or inside the tissues or organs that they affect. Some of these that you may see on the trees, such as the pictures down below, you'll see that oozing that can occur, as well as any fruits that can occur. You'll see this oozing coming out from the pores in that. But when bacterial pathogens are in the soil, they live mostly on plant material. survival is dependent on the decomposition of the material, soil temperatures, and moisture conditions. Some of the dissemination on bacterial is from plant to plant. That's what you're generally going to see. A lot of water. If you have water or rain off or rain splashes or irrigation problems, that can also disseminate the pathogen from plant to plant. Wind-blown rain or direct contact with the host. Insects and other animals can also spread bacterial pathogens. Humans are really big on doing this. Our cultural practices, you need to be aware of sanitation concerns when you're using your tools. If you're going from one plant to another and you feel that there is a problem with one of the plants, sanitize your equipment and your hands prior to going from plant to plant. The bacterial pathogens generally enter through openings within the fruit or tree or plant itself, either through the stomata that regulates the CO2 of the plant, injuries that occur, any type of heavy rains that might occur, it's going to damage those leaves, or any brushing of the trees against leaves or branches against leaves are all avenues for this bacterial to enter. Talking a little bit about viruses. Generally, soil-borne viruses that affect vegetables are very few in number. Viruses do need a living tissue of a host in order to survive. Fungal vectors and nematodes can transmit them. Viruses generally overwinter in your perennial weeds, flowers, and other plants. So if you're doing your gardening and you don't get rid of those weeds, if there's a virus in there, there's a possibility of overwintering in there. Often the crop plants by surviving in the roots of those plants, so make sure you get them out if possible. In the spring, as the plant resumes growth, the virus grows with the plant and emerges in the top leaves, where it's picked up by insects and carried to other hosts. When we talk about the Tombus virus group, generally you're going to see things like mosaics as far as symptoms, stunted growth, chlorotic leaves, yellowing of the leaves, and deformation of the leaf tissue. Some of the examples that we're going to have here as far as the viruses, such as the cucumber mosaic, where you see kind of a stippling or mosaic pattern within the plant or leaves themselves. Squash mosaic, you see a really good example of that up on the top there. Tobacco ring spot, where you see those rings. Tomato spotted wilt on the tomato. So these are very good indicators. The cucumber mosaic, all the cucurbits are susceptible. It overwinters in weed roots, tubers, and underground organs. It's introduced in the field via insects and human interactions. The squash mosaic survives between vine crop plantings and weed hosts, seeds, and overwintering beetles, of all things. Tomato ring spot. This is vectored by a nematode called the dagger nematode, and it's more common in sandy soils, has a lot of hosts as far as plants, generally occurs because of poor sanitation or rubbing of affected plants against healthy plants. And spotted wilt of tomato is generally transmitted by thrips. Now, thrips generally don't overwinter in the Illinois, but they can overwinter in greenhouses. So if you do have a greenhouse or a high tunnel in your area, that's something to keep in mind. Other viruses, as far as the transmission, it's vegetative propagation, rhizomes, tubers, any type of grafting. It can be in the mother plant. It will be transmitted to the baby plants. Seeds, insects, nematodes, plant to plant, and human contact. This is just an example of one of the of how a virus is spread. So up in the upper left-hand corner is day 1, where you see that leaf on the left is turned brown. That shows the infection. After 3 days, on the top right-hand side, you're going to see where it has gone from that leaf into the stem system of the plant and has carried it all the way to the top. And within 25 days, that whole plant has been contaminated. So even after 3 to 5 days, that virus is ready to be transmitted to other plants. Some of the fungal pathogens that we need to talk about. There are more than 100,000 known fungal species that live on organic matter. Most of them help with decomposition. But more than 10,000 of those can cause diseases in plants. And there are 2 types that you should be familiar with. Obligate parasites, they need a host in order for them to survive. And non-obligate, they may require a host for part of their life cycle, but they can complete the cycle on dead organic matter. So these are two different types of fungus you need to be aware of how they can spread. Some of the fungal pathogens that you need to be aware of that are soil-borne. Fusarium. Fusarium wilt overwinters and survives for many years in the soil as spores and mycelium and on the outer surface of other plants such as weeds and crops. They penetrate the host roots. It doesn't cause harm, but it can get into the plant. Disease is favored by warm weather. Generally, there's no cure for this disease. Rhizoctonia causes a wide variety of diseases, including the damping off, seedling stem canker, root rots, blights on vegetables and fruits. Warm, wet conditions, rhizoctonia can cause an aerial or web blight, affecting some ornamental plants. It does not produce spores. It exists specifically as mycelium or hyphae, and they develop a hardened overwinter mass called a sclerotia, and that's how they overwinter. So when those germinate, it will produce vegetative threads or hyphae of the fungus that will attack the fiber and food crops. Verticillium, there are especially the wilt of tomatoes or potatoes. Both have many other hosts such as cucumbers, peppers, strawberries, etc. Wide variety of hosts. Considered a vascular wilt. It affects the xylem of the plant or the system that carries the water and sugars throughout the plant. It affects that system in the translocation of water. Now, verticillium can survive in the soil for 4 to 6 years. Crop rotation is required with non-host plants, such as cereals or grasses if possible. Sclerotinia generally develop hard, black, overwintering structures the size of a raisin, just like sclerotia, and they allowed them to survive in the soil in the plant debris for 5 or more years. They will release spores under cool, wet weather in the springtime. Last one is Plasmodiphora. This can remain in the soil as resting spores or dormant cysts for many years, 6 to 8 years if there's a presence of a host. They will germinate in the presence of crucifer root exudate. So this is going to be specifically for clubroot of crucifers. Talking about the fungal pathogens, most of them have the mycelia, as you see on the right-hand side. That's what I'm talking about, mycelia and hyphae. Most of these fungi are produced by means of spores, which is the reproductive body of that pathogen. Disseminate is almost always passive. Wind, water, insects. Some are forcible, so as they start to ripen, they will force those spores out into the wind. That's what I mean by forcible. The fungal pathogens the survival and performance are dependent on the temperature and the moisture or presence of water in the environment. They can withstand broader ranges of temperatures and moisture, such as low winter temperatures and high dry summers. They also require favorable temperatures in order for them to germinate. The mycelium survives only within a certain range of temperatures like -5 to 45 and in contact with moist surfaces, either inside or outside of the host. Some of the fungal pathogens you may see or have seen in your garden, such as the Fusarium wilt. It penetrates the host roots and enters the vascular system where it moves and multiplies. The host plant will develop wilting symptoms, such like you see on the lettuce in the upper left-hand corner. They appear when the fruit begins to mature. You'll see a lot of problems and start seeing a lot of problems on that. The lower yellow leaves will turn yellow, sometimes on only one side of the plant, which is very unique if you've ever seen that. Leaf and stem will follow afterwards. On the Rhizoctonia type of plants on the right-hand side, these are most common symptoms of damping off, root rot. They can cause rotting of storage organs and foliage blights or spots, especially near the ground. Verticillium. Generally, the older leaves turn yellow and they die and they drop off the plants without wilting. Shoot tips will wilt during the day and may curl upward. Lower stem tissue is darkened by the infection. So on the left-hand side, you'll see where you have the discoloration of the vascular tissue. That's a very good indication that you may have verticillum associated with your problem. Fruit production is going to be reduced. Another one is sclerotinia, which are the white molds. If you've ever seen this, it's very unique, very easy to identify. It attacks beans, carrots, tomatoes, lettuce, squash. It infects the stems and the fruits themselves. The white mold infects the fruit through the blossom end of the fruit. No resistant varieties are available at this time. The cucurbit clubroot of the crucifers. The best symptom of the disease that you see on this is clubbing of the host roots. You can have similar symptoms to root knot nematodes and gall, where you may have fungal or bacterial infections. This can be found worldwide in temperate zones and infects over 300 species of crucifers. The plants can be stunted and is generally localized in low, wet areas of the field. So they're not going to affect every plant, but just certain ones if you have a problem with it. So how do you take care of all these different problems? These IPM control measures, they're very difficult to know what's causing it. You may need to enlist the help of a plant diagnostic clinic or an experienced pathologist to help you. There is a website here for the University of Illinois Extension for our plant clinic. Feel free to check them out. You need to keep written records, as some of these can remain in the field for years. The use of seed selection and storage. Clean seeds need to have the removal of pathogens. Synthetic fungicides and insecticides, if you're organic, are going to be prohibited. Those that can be applied can include biological seed treatments, clays, and botanicals. We'll cover that in a minute. Sanitation techniques, we talked about how humans can spread this. You try and remove and destroy as much of the old plant debris as possible. Depending on the disease, you may need to remove some of the soil as well. Adjusting fertilizing and water so they're not extremely succulent during the period of infection will help reduce some of this. Keeping plant foliage and stem dry also creates an unfavorable environment for it. Resistant varieties, resistant to certain bacterial diseases, is probably one of the best ways to do that. But there are varying degrees of resistance that may be available depending on the plant species you want to grow. So for the IPM, the crop rotation is another one. Of course, everybody should do some sort of crop rotation associated with it. Rotation between 2 to 3 years will allow plant material to decompose. Longer rotations of 4 to 6 may be needed for some other plant diseases. You need to control the weeds and insects. We talked about that. Destruction of plant parts or refuge harboring the pathogen, and the use of biological controls. Biological controls are going to be the use of live antagonistic microorganisms of that disease. It doesn't eliminate the pathogen, but it can reduce the number of the pathogen and restrict its ability to produce diseases. So when we talk about the water molds, these water molds are difficult control once they're established. Use the IPM or the integrated pest management procedures we just finished talking about. They can spread quickly in the field. Your soil, if you want to try and remove or try and eliminate or decrease the amount of incidents that can occur, they need to be light and drain well and quickly. Remember, the standing water is what's going to be important for that. Some biocontrols for organic, you can use copper products, hydrogen dioxide, streptomyces and actinovate products, as well as trichoderma products. More types of biological products, as you see here. These will be available to you at a later date. You don't need to write them down. Some of the bacterial pathogens, again, they're difficult to control. Use of IPM is essential on these. Some of the biocontrols are going to be bacillus, such as Ballad Plus or Sonata. Any type of copper oxide, such as Nordox, or Rhapsody, Serenade, Sonata. These type of products will be good for organic. Virus management, again, IPM is essential. Try and control the insects that are in your fields, and try and eradicate the host plants for overwintering. You want to try and keep it out of that soil if possible. You can do seed treatment and crop rotation on these, but spraying with fungicides and bactericides is not effective in the control of virus diseases. So these can be very hard to get rid of. Fungal pathogens, use of integrated pest management, copper products, Streptomyces, Bacillus, Cerenae, again, Max and Sonata are very good for these fungal pathogens. So as of that, I'm going to give it over to James here, so he can start talking about the soil health and how that's going to help. Are there any questions at this point while he is getting ready? Go ahead, James, and I'll take the question. You go ahead and start. James? James?
Can you hear me?
Yes, we can hear you.
Oh, okay, sorry. Okay, so we're going to look at a few options that are available to us when it comes to managing. I hate to use the word control because you never get to eliminating these pathogens, but you can manage them to a point where the damage is not overly severe. So let's first of all look at the soil system itself. We will look at soil health for disease management, what cover crops and organic amendments we can do in the soil. But before we get into that, we want to talk about what is soil health. This is a new concept. The word soil health is now replacing the word soil quality, which is what we've been thinking about before. So soil health is now being viewed as the continued capacity of the soil to function as a vital living ecosystem to sustain plants, humans, and animals. In other words, we got a job to do. We gotta manage the soil properly so that it can sustain living things. We've previously been thinking that soil is an inert system, which it is not really. We want to start thinking of soil as a living entity. Living things are the ones that have healthy systems. And if we think of soil as a living system, then we will start taking care of it as if it was a live organism and stop treating it as an inert material. So living organisms in soil that are supposed to be They perform functions, that is, the living organisms perform functions that are required to produce food and fiber. The products that are produced by the organisms in the soil are actually used by the plants. Now, on my picture there, on my slide, you can see the bottom right hand showing a very good soil ecosystem where about 50% of the soil system has water and air in it. It should be like that. And then you have the mineral particles or the soil particles, and then the roots of the plant find their way through all those particles to grow. Meanwhile, you have bacteria that are chewing down on organic matter to produce the nutrients that the roots are going to consume. And bacteria are being fed on by bigger critters, like fungi or nematodes or protozoa, as you can see in the soil food web on the top left-hand corner there. And that web is sustained by that healthy soil. And because you have healthy soil, you get healthy plants. So with that concept explained, then a few of the things that I'm going to talk about will become clearer. So then understanding the soil health requires that we know the biological, the chemical, and physical properties of that soil. It is said that if you take a teaspoon of good quality soil, you probably be holding at the very low end 100 million individuals of bacteria, and that's very low. As you see in a later slide, which is this one, a characteristic of a healthy, well-functioning soil, and I'm not going to read all of that, you can read for yourself, should be, healthy soil should be one where there's plenty of active, helpful organisms. And then above that, you also see that a good soil should have water, should retain water, but that it also drains well. That means the excess water is allowed to leave. So you need to have those two elements amongst all the others for microorganisms to survive well. Yes, and this was a statement I was talking about, the one that is highlighted here, that in a teaspoon of soil you could have more than 7 billion individuals. That's in a good soil. And so these soil microorganisms, which exist in large numbers in the soil, are dependent on a carbon source for energy, which would be the plants. So there is a very good interaction between these microbes and the plants. All right? And then while the bacteria and protozoa can withstand soil disturbance, as we always do when we till the land, that's what we are doing. We are disturbing the soil. We are causing a tornado in the soil, so to speak. But they come back. Fungi and nematodes prefer less of the disturbed soils. They like undisturbed soils more. So bacteria, I'm not going to talk about what they are. What this slide here is telling you is that while bacteria live around the film of water that is surrounding the soil particles, they also heavily colonize the area on the surface of roots. You can see there that the rhizosphere, which is the region just between the soil and the root, that's the rhizosphere region, it's heavily colonized when there is a plant system around. The roots allow that many more microorganisms to colonize that area. And you're also aware that colonies of bacteria will multiply to double their number in about 20 minutes. This picture was taken as an illustration of what we are talking about. The green area with the grass can be called a cover crop, and we'll talk about what cover crops are in a second. But the point from the previous slide, the point that is being made here is, while the brown areas on either side of this green region are bare, meaning there's no rhizosphere regions being provided to the bacteria and other microorganisms, in this patch of grass which was left behind, which is the boundary between the two fields, you still have lots of greenness and lots of habitat that is provided for microorganisms. So instead of, and this is where cover crops come in, instead of leaving your ground bare ever, you should always have it covered so that you have a lot of good things happening, but one of them being that you are encouraging the survival and the thriving of microorganisms that we need. Fungi. Now, when I talk about soil fungi, remember that there are some that are good, there are some that are not good. Remember what Rolly just said, only 10% of them are injurious to our crops, but they are the ones we are busy running after, and we kill 90% of the good ones in the process. In nature, 90% of the fungi can actually take care of the 10%. But because we want a perfect system, we kill everything. That's what we do with our pesticides. So the highlighted thing there is that fungi do release enzymes that break down complex organisms, including some of their own. They can actually digest some of the fungi that mess our crops. In addition, they are also important decomposers. They convert some of the hard-to-digest materials into nutrients that can be used by plants. So remember, keep that in mind. We also have a group of fungi that are called mycorrhizal fungi. Myco means fungus, the word myco, and rhizo means root. So these are fungi that associate with the roots, and it's a mutual benefit, and even more specifically, a symbiotic relationship, because the fungus is getting nutrients from the plant, or for the purpose of this talk, it's getting carbon for its own development, whereas the plant is getting a better ecosystem down there, which is now full of nutrients that have been availed by the presence of the fungi around the roots. Okay, now we already talked about fungi growing better in no-till conditions or undisturbed ground. And there's another group of arbuscular fungi that we won't talk about so much right now, but that they store or recycle nitrogen, and there will be one example later on coming. Organic matter is always important in the soil for a variety of good reasons, which you are familiar with. When you have organic matter, it's a source of nutrients, pure and simple. And organic matter acts as a sponge. Studies done by Rodale Institute show that in drought years, land that has been amended with compost or fertilizer or manure holds moisture a little bit longer than one that has been amended with the synthetic fertilizers. So it's a good thing to have. And compost also amends the soil. If you have very clay soil that is so compact and all that, if you add compost, it will improve the soil tilth. What's tilth? The physical nature of the soil. So when you have very compact ground, you have very poor tilth, and roots of crops will have a very difficult time penetrating that. So all those good things about organic matter, which is either alive or dead, and which becomes available to other microorganisms for breakdown into nutrients. So let's talk about cover crops. A cover crop is one that covers bare ground. You know that Mother Nature never leaves any ground bare. She always covers it. Same thing that we should be doing. We should never leave ground uncovered, like between seasons. And therefore, farming systems are now going into that practice of never leaving the soil bare between growing seasons. So this is a crop you introduce in between the seasons. You don't wait until you harvested everything. You can actually, these days, they have ways of incorporating cover crops into the existing crop just before it's harvested. You can do that too with your backyard gardens, and this would help with not leaving the ground bare. Without reading everything, you read all the benefits there, but the one in red says the cover crops may suppress disease resistance. So in addition to, the cover crops help with the disease suppression because some of them actually produce fumigants that are injurious to pathogens, disease-causing organisms. And we'll be talking about them in a little while. I'm not going to read everything here, but suffice it to say that some have been shown to reduce the soil-borne pathogen inoculum in the soil. What are the features of an ideal cover crop? Seed is easy to, well, the cheaper the seed, the better. If you can just right now buy annual ryegrass and sprinkle it on your backyard garden, that's a very cheap that's a very cheap cover crop. Easily available, soybean. Soybean is another easily available seed that you can just grow. Maybe late now for us in the north to grow that one, but annual ryegrass is king when it comes to these types of weather. And then easy to establish and fixes, captures nitrogen. That would be a bonus if you can do that. The deeper the roots, the better the opening of the ground. If you can inhibit weeds, even better. You don't want it to become a weed itself. So even again, that's another good thing. And then spring planting is not interfered with. And eventually, you get a yield of the primary crop. Here is a good example of a cover crop that has been shown to confer some advantage to soils that have been infested with nematodes. And the comparison here is between no cover and a mustard crop cover and an oil radish cover crop. And you notice that with the oil radish, the blue column there, which indicates non-parasite nematodes. They are nematodes, but they are beneficial. Increased maybe 3-4 times as much as the population that had no cover, or the one with the mustard. In other words, the oil radish must be doing something in the soil to encourage or to induce bigger populations of the beneficial parasites, the ones that will now, beneficial nematodes that will now predate on the ones that are dead. So how do the cover crops achieve this disease suppression? First of all, you are extending the crop rotation. So if you're growing maize and then you want to grow soybean thereafter, in between, if you can have some sort of like cereal rye, it's a non-host of the pathogen that may get into soybean. Therefore, you've just extended the length of the crop rotation. By having that non-host, you are reducing the inoculum levels of the pathogen that would have otherwise caused problems for the subsequent crop. Then when you improve the soil structure, as we just discussed a while ago, air movement. By the way, remember that air moves freely in and out of soil. Air movement is improved when you have all those tunnels and channels that the roots make. And then porosity, movement of water, is improved. And then there's a physical barrier if you have some of these cover crops, so you don't have weeds growing as much as they would have otherwise. And these weeds could also have been hosts to some of these pathogens. And then you now stimulate microorganisms to grow more, like the bacteria or like the nematodes we just saw. And then some cover crops produce biofumigants. And at this point, what's biofumigation? Those of you that may know about nematodes in soil and know about chemicals may recall something called methyl bromide that is actually injected into the soil and held in there to kill nematodes. A similar gas is produced by brassicas and mustards by an enzyme action and is effective also at killing nematodes. So isocyanate is a gas that is produced by these crops that can actually kill nematodes. So brassicas and mustards, when they're used as biofumigants, they are annuals, of course. They can be used as annuals, grown in winter or spring, and they play many other roles as cover crops, not just biofumigation. And those examples are shown there. And radish, a picture of radish growing there that is used as a cover crop, you're seeing it there. It's very aggressive in its growth. You see it heaving out of the ground because it hit a compact end down there. So it's growing outwards as opposed to growing downward, just because it's either you take the path of least resistance, which is upwards. So this one variety of mustard, or called Kodiak mustard, is supposedly very good at suppressing soil-borne pathogens. And in the next slide, I give you a procedure for incorporation of these biofumigant crops into the soil. It's not easy, but if well done, it's supposedly very highly effective. What's the best time to seed? Anytime, between April and September, depending on the rotation. Are you a field crop grower, vegetable grower, whatever your system is like, you can always incorporate this mustard at any one time. Seed rate per hectare or per acre is given in both. Both of them are given here. If you have a heavy crop, a heavy soil, you overseed. If it's light soil like sandy soil, then you plant less. You add some fertilizer, because even cover crops are crops that you need to grow and thrive with vigor. And then the soil preparation, as you would any other. Sometimes you don't have to do it. When they seed in some of the annual cereal rye, or even the clover, red clover, they don't really prepare the ground. You just let it rest on the ground, and between freeze drying and all that, you get them incorporating pretty well. Otherwise, you can just seed in with a grain drill if you have prepared the ground like you would for a normal field crop. And then, once this plant, the mustard is growing, and you estimate it to be at 60 to 8% flowering, you chop it down as with a lawnmower. That would work too. But then between chopping and incorporating should not be more than 30 minutes. The longer you wait, the faster you start losing the isothiocyanates that are being produced as a result of the injury of the crop. So and then if you can manage to cover that area with a plastic, clear plastic, fine, if it's not a big garden. If it's a bigger garden, if you can irrigate with just a thin layer of water, 10 millimeter is adequate, that will hold the very highly volatile isothiocyanate fumigant in the soil to do its job. Then you work the field 2 weeks after incorporation, but you can't plant anything until 3 weeks after the incorporation. So you have to wait a little while. Everything has its disadvantage. This is one disadvantage. You have to wait quite a bit. So that's that about cover crops. We talk about composts now, and composts have the potential to also reduce the amount of soil-borne pathogens in the soil. First of all, compost provides vigor and health to a plant. A well-growing plant has the capability to withstand infections and infestations by insects. So that helps quite a bit. The only thing that, and because of time I have to rush, the only thing about compost is that all composts are not created equal. If you use compost that's just about finished composting, it's devoid of advantageous or useful microorganisms. That means you have to wait for it to cure to regain that population of the microorganisms that it should take into the soil. So you need to cure it for a little while longer, maybe another 2 months, to get the goodness of it. Okay. And there is a slide here showing the ability of manure and compost to reduce the pathogen load due to cucumber mosaic virus as well as septoria leaf spot, but not in anthracnose. Not everything works as would be expected. But you can see there that manure did reduce the pathogen load quite a bit compared to compost, but both of them, where they worked, did work better than when neither was applied. You can modify the environment. We talked about water drainage, so don't over-irrigate. In fact, if you can do drip irrigation, the better. It's also been shown that if you have too many seeds when you are sowing chili peppers and you have too many seeds in the container that you are sowing it in, then you get stress out of the population and you have more of dumping off happening. And mulching, like you all know, prevents splashing off of soil-borne pathogens onto fruit like watermelons or cantaloupes. If you can, whoever is selling you seed, ensure that they have certified disease-free or pathogen-free seed. They check them out. They know how to check them. Indeed, also this goes for plants. You're buying strawberries to go and transplant. Ensure that whoever is selling it to you checked them out, not selling you something that once you have virus in the system, it's there. Or you don't want to start treating them in the first place. You can also do hot water seed treatment, and it's shown in this slide here by Sally Miller of Ohio State University. Different seeds can be treated to various temperatures of water for various periods. You can see a little caveat there that cucurbit seeds, if you have those types of temperatures shown there, you may cook them and that way they lose their viability. So there are all sorts of things. This is something else you can do. If you can find resistance, which is genetic, a plant and a pathogen interact, that we know. And the genes of the pathogen provide this toxin, and the genes of a plant recognize this toxin, and they can either break it down or counteract it in some way. And it's always a race of who can overpower the other. If the pathogen is overpowering, if it's virulent, as we say, then the plant succumbs. On the other hand, if this pathogen comes in and it is not able to overpower the genetic mechanism of the plant prevails. Now, that's the best and easiest for the environment. For us, all you have to do is get your crop going and you're good. However, remember that pathogens are always mutating. There's always mutations, and you only need 1 in a billion of those pathogens to mutate to a DNA that can overcome the plant resistance. And we get back to breeding again. Okay, and resistance is not available for all diseases. Grafting. This has been shown very well to work for soil-borne pathogens. Indeed, I'll go back to one that was mentioned by Rory, Ralstonia solanacearum, bacterial wilt of tomatoes. If you can get a variety that has resistance, and for those of you that love the heirloom tomatoes and graft the two, the heirloom tomato may have susceptibility. If you graft those two, the rootstock will provide the resistance to the above ground portion, which is the scion. So again, this is a very nice and easy way of doing it. If you have a very ideal system where the ground there, as you see, has compost, it has a cover crop of sorts that you use, and microbes are happily growing there, and you have plant growth promoting rhizobacteria, the plant may well be induced to do something akin to immunity. Like us, when we are exposed to a pathogen, we do gain antibodies. It's almost similar, not quite, but like that. So that the plant will get what is called the systemic acquired resistance. Why systemic? Because whatever we call them phytoalexins in plants, the antibodies, or if you have the salicylic acid now being produced as a result of infection, you have a systemic acquired resistance that is now going throughout the plant. So that can happen too. Studies are still going on. It's a very complex subject. The mechanism is not very well known, so I won't say much more beyond that. Next, you can actually inoculate mycorrhiza into the soil. We talked about mycorrhiza back there. Myco is fungus, rhizo is a root. If you can have more of this, you end up with a root system that is larger and more active. You have a plant that will tolerate root pathogens better, and you reduce, because of competition between these fungi and the bed fungi or other microorganisms, you have reduced, you have competition for resources, and you outcompete the bed guys. All right, and then we may very well increase host resistance, as in that systemic acquired resistance we just saw. So, in summary, if you do a whole lot of things, so there is no one, first of all, you've already noticed that there is no silver bullet here. Nothing will solve the problem here, one single item. IPM is the way to go. You're doing a whole lot of things above ground and more below ground. If you have healthy soil, you have healthy plants, you have healthy growth and healthy production. And a few references for you to read, which you can't copy now, but if you ask for these handouts, we'll send them over.